got these chromosomes and genes, and they're made up of letters. And so a genetic change can be just a change in a single letter of this code that happens during DNA replication. And so a lot of the changes we see, which are called mutations or variants or changes, can be at the very small scale. So just a single letter in this code. And so this is an example of what DNA effectively looks like. There's 4 letters, A, C, G, and T, and DNA is just a string of those. It's a very long string. So it's 3,000 million letters. And one of my more cultured colleagues said that's the same number of letters as you'd find if you read War and Peace 1,000 times. So pile up 1,000 copies of War and Peace, that's 3,000 million letters, okay? That's a lot. I'm a little less cultured. I tend to go for the Harry Potter analogy where it's 5,000 copies, and we'll get onto that. But so you've got this string of letters, and the simple changes that are called point mutations or sometimes single nucleotide variants, SNVs, it's just one letter changes, from a T to a C, and that may or may not cause a problem. But so they're the most common causes, those sort of changes of inherited disease. But you can also have much larger changes affecting, tens, hundreds, thousands, even, many thousands of the letters. And so here's showing like a nuclear gene chromosome, and there's 6 regions, A, B, C, D, and F. And in this case, we're seeing, one of the Ds has gone. It's been deleted, so that's a deletion of a substantial chunk of the DNA. You can also have kind of the inverse. Instead of A, B, C, D, E, F, now A, B, B, C, D, E, F. So there's a duplication. And again, those may or may not be problems. And I mentioned there's, 3 billion letters. If one has whole genome sequencing, out of those 3 billion letters, any individual will have about 1 million of these changes, the point mutations. And so clearly most of those are not going to be causing disease. They're just variation. And our challenge is to distinguish, which are the meaningful ones. And then in terms of these larger scale changes, any individual will have a couple of thousand changes of those sort of structural variants. So, when we do this large scale sequencing, we find a lot of stuff. And so the challenge is interpreting it. So here's my Harry Potter analogy. So 3 billion letters, it's about how many you'd find if Harry Potter had 5,000 volumes instead of 7. And just to labour this point, I guess, if you look at the first sentence of the first book, Mr. and Mrs. Dursley of number 4 Privet Drive were proud to say that they were perfectly normal, thank you very much. And so a single nucleotide variant or a point mutation is just something like this. One letter has changed in the sentence. It's now Thursley rather than Dursley. That doesn't have a big impact on this sentence, probably, other than, it looks a bit funny. And so it may or may not be a problem. And then you can have these small changes where you get a string of letters that are deleted. So what's that, 2, 4, 6, 8, 10 or so. And so if you're trying to read that, then they were proud to say that they were permal. Thank you very much, which is, a little bit odd, may or may not be a big problem. Depends how important the sentence is. I guess that first sentence is always pretty important to get you in. But then, instead of one of those letters changing, this time it's been deleted. And so we've lost the R. And when you're reading DNA, typically, if you lose one letter, everything else just gets read in sort of that same frame and can therefore not make much sense. So instead of, living at number 4 Privet Drive, it becomes, Numbeth, Alp, right? So those words no longer make sense. And those sort of changes, it's only a single letter that's changed, but it just messes up that sentence completely, or maybe a whole series of sentences. Just trying to emphasize that there are different types of changes. You have funny ones that can be quite hard to detect with some of the sequencing technology, where you take a chunk out of the sentence, flip it around and put it in the other way somewhere else. So the sentence becomes, so it's just, it's complete gibberish. Or you could have a bigger change. So maybe in the, halfway through that first sentence of the novel, going to pretty much the last page of that volume. And so, you've got a huge amount missing, and in terms of genes, that could be many genes. Sometimes we get this loss of, 3, 10, 100 genes. And those sort of changes tend to be important because at least some of the genes are going to be meaningful. So just trying to emphasize that, there are all these different types of changes, and that's obviously going to, and there's lots of them, so that's obviously going to make it difficult to determine which ones are meaningful. So here's an example in terms of mitochondrial DNA with these small scale changes, and some of the terminology that many of you will be very familiar with, like in MELAS, the most common change is called M for mitochondrial DNA, M3243A to G, so that's a one-letter change. Similarly, most common mutation in MERF, M8344A to G, again, a single letter change, and likewise in NARP, or maternally inherited Leigh syndrome, and Leber's hereditary neuropathy. They're one single letter changing, and typically, they can either change a protein or interfere with how well the proteins are made. And then you can get large scale changes of hundreds, thousands of letters. And so in mitochondrial DNA terms, these are the classic causes: deletions of the mitochondrial DNA, large chunks that result in conditions such as Kearns-Sayre syndrome, Pearson syndrome, and progressive external ophthalmoplegia. Mitochondrial DNA was a whole new world, and it was back in 1988 when changes in the mitochondrial DNA were first shown to cause disease. And so, in eggs, there might be 200,000, 500,000 copies of the mitochondrial DNA. In most cells, it's more like about 2,000 copies maybe. And so, as Emma said, it's not 0, 1, or 2 copies of the gene that are functioning or not functioning. It could be, 0%, 5%, 20%, 70%, or 100% of the mitochondrial DNA that's changed. So in this diagram, it uses the scientific nomenclature, if you like, for a wild type cell, and that's wild, and that's sort of, hey, I'm feeling pretty wild. So this is what normal. Wild type means normal. It's just genetics, literature. Expect nomenclature to try to keep other people out of the field instead of saying healthy or normal or whatever. So wild type is what we call healthy/normal, and this is called homoplasmy when all the mitochondrial DNAs have the same sequence, no changes there. And that's homoplasmic, healthy, if you like, or wild type. And the other extreme, which is quite common in, for example, Leber's hereditary neuropathy, is that every mitochondrial DNA in the cell and in the body has been changed. So this is now homoplasmic, or what we call a pathogenic variant, meaning something that causes pathology. So it's a disease-causing variant. And for most conditions, that status means that someone is very likely to develop disease. So the challenge then becomes when you have these intermediate levels of disease. So for a lot of mutations, they can be present at maybe 20%, 30%, some of them even 80%, or more of the DNA can be mutated, if you like, or disease-causing, and they're still unlikely to cause disease. And for common mutations, we have a pretty good feel for how much is needed before it causes disease. But somewhere along that range from 20, 30% up to 90% or whatever, you might hit a threshold, a clinical threshold, where that mutation is now going to cause enough problems in the cell that it stops working well. The challenge is that actually picking that can be quite difficult, particularly for rare mutations. And then it opens up these areas that are complex because heteroplasmy can differ between tissues. So it's well known for some mutations, as in MELAS, at birth, it seems like the level of the mutation is the same in pretty much all your tissues. But then in blood, you tend to lose the mutation at maybe 1 to 2% a year. And so testing someone when they're 50, the mutation may not be visible, at least using the old-fashioned technologies. Probably for the point mutations now with the new next generation sequencing, we can detect those sort of levels. But certainly for deletions, for example, they can be absent from blood. So with almost all other inherited diseases, it doesn't really matter whether you test blood or saliva or muscle or whatever. But for mitochondrial DNA disease, that can be important. And so, the people doing the testing need to understand those limitations. So again, good to emphasise that mito disease can be caused by changes in either the nuclear or the mitochondrial DNA. And the rule of thumb is that for children, for pediatric onset, so onset in childhood, roughly 3/4 of the time we find the cause is in one of the nuclear genes, whereas in adults it's about the other way around. And so for adult onset disease or late adolescence, it tends to be maybe 2/3, 75% caused by mitochondrial DNA changes.